In this video, we're going to talk about the location of DNA in eukaryotic cells. So let's do a review of what you learned in freshman biology and make sure we're feeling good about the idea of a eukaryotic cell versus a prokaryotic cell. Eukaryote is you. You are made of eukaryotic cells. So humans have eukaryotic cells because they have a nucleus, which is what this large organelle, this blue organelle is right there. So eukaryote has a nucleus. The other thing that eukaryotes have is that they have lots of organelles made of membranes. So they have membrane-bound organelles. So if you remember back to bio, you probably remembered some of the name of these. Things like mitochondria, and endoplasmic reticulum, and Golgi apparatus, lysosomes, vesicles, things like that. Eukaryotes have lots of different parts to do different jobs. Prokaryotes, on the other hand, think early. These are the early types of cells. Prokaryotes have none of those things. They don't have a nucleus, and they don't have membrane-bound organelles. Their DNA is found in something called the nucleoid, which basically means that the DNA is free-floating. It's just floating around in the cytoplasm. They don't have organelles with membranes, no mitochondria, no chloroplast, no endoplasmic reticulum. Pretty much the only organelle they got is ribosomes. Ribosomes, we're going to learn a lot about them this year, are the protein factories. They're what make proteins. Okay? So every cell needs to have DNA and proteins. It's just, how complicated are they? Do they have lots of extra little pieces to do different jobs? So little organelles. Okay? You are complicated. You have eukaryotic cells with nuclei, organelles. All right. So why is it good that you have a nucleus in every cell in your body? Well, a nucleus allows for compartmentalization. So throughout your cells, there are different organelles for different jobs, and that's going to allow you to do things more efficiently than a bacteria cell can. And because your cells are able to do different jobs, they can work together to make a big, complicated organism like you work efficiently. Also, DNA is housed in this basically nuclear membrane. That's what we see here, this gray outer layer. Okay, so this is our nucleus, and imagine like we cut it and we took like a chunk out of it, and you're seeing inside. So you see this nuclear membrane, okay, also called the nuclear envelope, and its job is to protect all this DNA inside. DNA is called chromatin. It's another name for it. It's basically the DNA and the proteins that wrap around it to help fit it into the cell. Our cells also have something called the nucleolus, which is kind of a dark spot in the nucleus. And that is the part of the DNA that actually makes ribosomes. Okay? And so that's found in there. But the main thing you need to remember is it has this special membrane surrounding the DNA, and that's going to protect it. And it protects the DNA so that there's fewer mutations due to environmental damage. Okay? Back here, our prokaryote, its DNA is floating around. Let's say we have some UV light striking that DNA is going to have a mutation, whereas the nucleus can deflect some of that UV light, so there's less mutation. So that's going to be really helpful. Fewer mutations means you can have a constant species. You're able to pass traits from generation, good traits from generation to generation. You're not making as many potentially bad traits that might harm or kill the organism. So how did the nucleus actually come about? Well, scientists think that the cell membrane actually just kind of started to fold in. Okay, and so here you can see we have these infolding of the cell membrane, also called the plasma membrane, and it kind of starts to wrap in and wrap around the DNA that's free floating, and it forms a nucleus. Okay, and you can see that this kind of infolding of the membranes also created lots of other organelles, like the endoplasmic reticulum, also called the ER, maybe you remember that from bio, or the Golgi apparatus, Golgi bodies, things like that. Notice mitochondria and chloroplasts right here. So here's the chloroplast, here's mitochondria. They evolved differently. So let's look at mitochondria in a little bit more detail. So how exactly did mitochondria evolve? Well, mitochondria, scientists believed, evolved through something called the endosymbiotic theory. So notice the word theory. So this isn't just some hypothesis. There's a lot of evidence to support this idea, and it is generally accepted as how eukaryotic cells came about. So in the past, there was this really simple kind of eukaryotic cell, okay? There was infolding, it had a membrane around its DNA, so it had a nucleus, it had some endoplasmic reticulum and things like that. So it was a really basic kind of cell, and we call that the host cell. And this host cell can engulf other cells, and they would do this, and they would basically release enzymes and digest those cells and essentially eat them. Amoebas do that all the time. But something happened in that rather than eating this little cell that it engulfed, they lived together in symbiosis. So if you remember back to biology, symbiosis is basically a really close relationship between two organisms. They depend on each other by living on or in each other for basically survival. So in this case, this little prokaryote got engulfed by this host cell, and they lived together. And they were so beneficial that eventually the prokaryote became so dependent on their host, they became organelles. So this little prokaryote that got engulfed, it had a special home. It had extra protection from that cell membrane and things like that. And this big host cell was providing it with energy molecules like food. And what the little prokaryote would do in exchange was it'd break down that food and produce ATP, which is what mitochondria do. They're the powerhouse of the cells. So this prokaryote became so dependent on their host that it became organelles. Same thing would have happened with a photosynthetic prokaryote. It could have been engulfed and lived in symbiosis, and it would have become the chloroplasts. Humans don't have chloroplasts, and in this class we focus a lot on human genetics, so we're not going to focus on that. We're going to focus on the mitochondria. But basically, this prokaryote that could have done cellular respiration, okay, and produce ATP from food, that's what's going to become the mitochondria. So here you can see it in a nutshell. Okay, you have these two independent cells. One engulfs the other. It lives inside. They benefit from each other so much that eventually they start to just be passed from one generation to the next. Now, why do we care about this? Okay, so why do we care about mitochondria being engulfed bacteria? Okay, well, it means that in humans, the nucleus isn't the only place that has DNA. Okay, mitochondria also have DNA. They have circular rings, just like bacteria do. If we jump way back to this picture, okay, eukaryotic chromosomes are linear, and when they're duplicated, they look like X's. Okay? This prokaryote basically has a big ring of DNA. It has circular chromosomes, not linear chromosomes. So the fact that mitochondria have these little rings of DNA means, hey, they're more like a prokaryote. They have DNA like prokaryotes, so that's evidence for it. And so mitochondria have their own little rings of DNA. They have about 37 genes on them. And those genes tell the mitochondria basically how to make proteins to do cellular respiration, how to be the powerhouse for the cell. Okay? Now, how exactly is mitochondrial DNA, which we abbreviate as mtDNA, how exactly is it inherited? Well, we inherit our mitochondrial DNA from our mothers. Okay? So, when the egg is fertilized that became you, okay, the sperm from the dad only contributes a nucleus. So here you can see the dad's DNA going into the nucleus. The mom, on the other hand, the egg that she created not only contributes half the nucleus, but the big egg also contributes all the organelles, including the mitochondria. So all the mitochondria in your body originated from that egg cell that came from your mother. So why do we care about mitochondrial DNA? Well, mitochondrial DNA allows us to trace maternal ancestry. Now, DNA from the nucleus, it's really cool in the fact that it's a blend of both your parents. You get DNA from your mom and from your dad in the nucleus. But that can be really hard to track, right? Because you're blending families along the way. But with mitochondrial DNA, because it only comes from your mom, it allows you basically to trace more accurately your ancestral history. Okay? Because you're only tracing direct passage of mitochondria from mother to all of her children, both male and female. And this is really cool because mitochondrial DNA has been used in all kinds of different ancestry experiments, including human migration. So scientists used mitochondrial DNA to basically trace and figure out the origins of humans. So scientists theorize that the first humans were in Africa, and through tracing mitochondrial DNA, they found that some humans stayed in Africa, others began to migrate to different parts of the world, including Europe, the Middle East, Asia, and then crossing over through Alaska, they were able to go into North America and then South America. So they can look at the genes in the mitochondrial DNA and actually trace the ancestors and trace the lineage throughout human history. It's pretty fascinating. So in this set of experiments, or this week, we're going to be learning a lot more about mitochondria, how they came about having their DNA, and some cool experiments that we can do with mitochondrial DNA.